So yesterday we finished talking about the lymphatic system and how the molecules in the lymphatic system combat pathogens within the body. And so this is just a picture of the lymphatic system. And it travels, and it works in communion with your immune system. Antimicrobial peptides and proteins. Interferons interfere with viral defenses. They help fight the cell-to-cell spread of a virus so it doesn't go to other parts of the body. And other interferons are used to activate macrophages to combat other viruses or pathogens. The complement system consists of roughly 30 proteins in the blood plasma. These constantly circulate in an inactive state, and they are activated on the surface of microbes. And then they kill the cell by biochemical reactions, and this is one of the, this process leads to inflammation. Your inflammatory response. Histamine cells are released, triggering nearby vessels to dilate and become more permeable. Capillaries leak fluid, causing the swelling. Also, once macrophages and neutrophils are activated, they just discharge cytokines. And cytokines are signaling molecules that control immune responses. Cytokines promote blood flow to the area. That's why it's red and swelly, and sometimes it gets heated up. And then this is just a diagram explaining the inflammatory response. And as you can see, so like if like a splinter gets into the body, the macrophages, they go and they attack the bacteria or the pathogen, and then they consume it and take it away. And then neutrophils go to the area, and this causes swelling to stop the spread of the bacteria.
Well, the swelling just allows white blood cells to escape into the tissues. So the whole idea of the inflammatory response is just simply mast cells releasing histamines that makes the blood vessels dilate, that allows fluids to get out, increases blood flow to the area, and also allows white blood cells to get into the surrounding tissues in a process called diapedesis, where they squeeze between endothelial cells of your blood vessels and get into the area where they can actually ingest the invading microbes.
Yeah, everyone got that? Yeah. And then, like, the pus you see after a cut is a result of the dead pathogens
and white blood cells.
Yeah, and like cell debris. Inflammatory responses. Massive injuries create systematic responses. That means like your whole entire body gets involved to send stuff to the big injury to attack the bacteria there. Fevers also are said to stop fighting infections because it's hypothesized that it increases the cell response to the bacteria into the cells. And then, septic shock occurs when bacterial infections induce an overwhelming inflammatory response. That is bad, and it's fatal in more than 1/3 of the cases. And this is accompanied by high fever, low blood sugar or blood pressure, and poor blood flow. Also, Crohn's disease and ulcerative colitis is another disease involving the inflammatory system. It actually involves the intestines, and it's where the inflammatory response, overworks and is counterproductive. The evolution of pathogens. Some pathogens have evolved to avoid destruction by these cells, and tuberculosis is an example of this. The bacteria M. tuberculosis, that obviously causes tuberculosis, is engulfed by macrophages, but instead of dying within the macrophages, they just they keep reproducing, and then the tuberculosis never goes away. That's why it's one of like leading causes of death in the United States. Adaptive responses. These rely on T cells and B cells, and these kinds of cells are types of lymphocytes. The B cells originate in the bone marrow, so you can obviously remember that by B, bone marrow, B cells. And then others migrate to the thymus, or T cells. You can also remember that, thymus T. And then the antigens, they elicit T cells and B cells to the area. Recognition occurs when B and T cells bind to an antigen, or also known like as an antigen receptor. Does anyone have questions on that? I'm going to talk more about it. All antigen receptors made by T and B cells are exactly identical. These contain about 100,000 receptors, which coincide with a specific pathogen. And then an epitope
Yeah.
Epitope is the accessible portion of an antigen that binds to an antigen receptor.
I'm going to give you an analogy for that. Epitopes are like the mugshot and fingerprints of the antigen. So when the immune system needs to recognize it, the antigen, let's say for future encounters, for immunologic memory, okay, so that you're immune. It doesn't need to remember the whole antigen, it just needs to remember certain bits and pieces of it that make it identifiable. So the identifiable parts are called the epitopes. And that's what the antibodies in the humoral response are made against. They're made to match the epitopes so that the antibodies combine with only the epitopes. Some antigens have thousands of epitopes. Some have only a few, So because anything foreign to the body is considered an antigen. Anything that causes an immune response when exposed to the body is called an antigen by definition. So an epitope is just its identifiable parts, kind of like the mugshot and fingerprints. Yeah. If you took a picture of somebody's torso, how are you going to identify somebody by that? You see what I'm saying?
Yeah, tattoo.
Well, but that tattoo then would be then an epitope if that person was an antigen.
Okay. Okay, so now I'm going to talk about specific B cell and T cell recognition. B cells are Y-shaped, consisting of 4 polypeptide chains, 2 heavy chains and 2 light chains linked together by a disulfide. And then right here, this is an example of a B cell. So you can see there's the 2 heavy chains, you have a light chain here, and then these 2 heavy chains are connected by a disulfide bridge.
Now, the other thing too on that is the chains are also just a name for the polypeptides. There's 4 tertiary structures you're looking at there. 2 light ones and 2 very heavy ones in the middle.
Okay, so B cell activation. So when a pathogen enters a body, you can see that these are the antigens right here. And then on top of the antigens are the epitopes. And then the epitopes are what bind to the cells. And the antigen receptors, they bind to the antigen receptors to disable the cell, and so it doesn't reproduce anymore. As you can see, right now it's getting disabled. Does anyone have any questions on that?
Think of antibodies as being handcuffs. It doesn't destroy the suspect, but when it binds to the suspect on the wrist, it neutralizes the suspect, doesn't it?
Yeah.
Doesn't kill him, okay? Just neutralize. And that's what antibodies do. When they bind to an antigen, they simply take away the threat. They haven't destroyed it yet. That's up to a macrophage to do later.
Okay, and then this is it. This is just a T cell. You see how it's different, obviously, from the
So what's that thing in the middle?
this thing,
Mm-hmm.
the disulfide bridge. So that's similar in both the B cell and the T cell.
Right, but what is that whole purple structure there?
Oh, a polypeptide chain. Is that what you mean?
Yeah, but what is that specifically?
I'm not sure.
Okay, that thing, and you see that it looks like spikes all over the cell?
Yeah.
That is called the major histocompatibility complex.
Oh, yeah, I covered
Or protein.
That. Yeah.
Yeah.
I covered that. Yeah.
Okay.
Yeah.
Oh, there it is.
Right here. Okay, so T cell activation. Unlike B cells, which bind to antigen on the surface of the pathogen, the T cell only binds to fragments within a pathogen, and these they only bind to antigens that like are displayed on like the surface of a host cell. I'll show you a diagram later, or next slide, to help explain it. And then the major histocompatibility complex are the proteins that display the antigen. So as you can see here in this diagram, the T cells, they're only going to bind to the antigens that are displayed by the MHC from the pathogen. And this disables the pathogen just like the B cell. Does anyone have questions on that? Characteristics of adaptive immunity. Immense diversity of lymphocyte receptors. Self-tolerance. So that means it's not going to attack your good cells, it's only going to attack the bad cells. Cell proliferation increases T and B cells, which means the more cells that are created, the more T and B cells are going to be. More rapid response to already discovered pathogens. So once a pathogen enters your body, if another time it tries to enter again, it's going to be, your immune system is going to work harder to prevent it, and it's going to be easier for your immune system to combat it. Immunological memory. Your primary immune system is first responds in 10 to 17 days, and that's just to fight the pathogen that's in your body. Your secondary immune system is the immune system that works hard to withstand the pathogen if it ever tries to enter your body again.
So the defenses provided by the B and T lymphocytes can be divided into the humoral immune response and the cell-mediated response.
Cell-mediated.
So the humoral immune response involves the activation and the cloning of the B cells, and it results in the production of secreted antibodies that neutralize or eliminate toxins and pathogens, and that circulates into the blood and the lymph, which was once known as the body humor fluids.
Right. Humors is a very old term that refers to the fluids of the body. So it's a fluid immunity.
Yeah.
You got an immune system that's mediated by cells, cytotoxic immunity, or cellular, and then the ones that are mediated by antibodies dissolved in serum, the fluid immunity.
Involves the activation and the cloning of the cytotic T cells, which directly destroy the infected host cells, unlike the other response. And so, to do this, they need the helper T cell, and that triggers both the humoral and the cell-mediated. What is the word?
Mediated means it brings it about.
Okay.
It makes it happen.
The cell mediated immune response, but it does not carry out the responses. And the helper T cell just sends the cell signals that initiate the production of antibodies that will neutralize and destroy the pathogens and activate T cells that will kill the infected cell if needed. There are two requirements for the helper T cell to activate the adaptive immune response. And the first one is the foreign molecule must be present that can bind specifically to the antigen receptor of the T cell, which was kind of like what Emily was saying earlier, that it can't work unless they bind specifically. But there's many different types of T cells that they'll find one to bind to it. And then the second part is the antigen must be displayed on the surface of an antigen presenting cell, and that is a cell that can be a dendritic cell, a macrophage, or a B cell, the antigen presenting cell, and that plays a big role in the responses for the rest of this section.
Okay, so folks, just to kind of make sure that you guys understand this, any of the phagocytic cells, macrophages, dendritic cells, just like Maggie said, their job is to engulf things that they think is foreign, right? Mm-hmm. But once they do, if they present the epitopes to a T helper cell, now their title changes to antigen presenting cell, or APC.
When host cells are infected, they also display antigens on their surface just like the antigen presenting cell. And so the way you can decipher from the two is the MHC molecules, which Emily just talked about, because most body cells only have class 1 MHC cell, but the antigen presenting cells have class 1 and 2, which provide the molecular signature, which is also the epitope, to recognize that it's the antigen presenting cell and not just a regular body cell.
Yeah, so in other words, only antigen presenting cells carry the MHC2. Mm-hmm. I use the analogy of like a cellular badge. Everybody's got the driver's license, including the police. But the police have something we don't have. An extra badge, an extra ID that we don't have. Do you follow what I'm saying? Mm-hmm. That's like their MHC class 2. The driver's license like the MHC class 1.
Right.
So the class 1 just identifies your cells as belonging to you. But the MHC class 2, that's their little cellular badge that tells the T helper, hey, I'm an antigen presenting cell. I'm presenting an epitope to you.
Mm-hmm. Did everyone understand that part? And then the helper T cell interacts with the antigen presenting cell. So, can I show this?
Sure.
This is just how they interact with each other. With that down, do I have to copy and paste it?
Probably. So what analogy could you use for the T helper cell then? If the antigen presenting cell is presenting the antigen to the T helper, and the T helper is then going to orchestrate cellular and humoral immune responses, what analogy could you use for a T helper cell?
Hold on 1 second. Why can't I?
Because it's like on the iPad, you can do it.
I can do it.
Oh, that's right. Since that is on the iPad,
Yeah.
you can't use the computer cursor to do that. You have to use the iPad.
Can I get a drink of water?
Sure. Emily, you'll have to prop the door open and then pull it shut when you come back in.
Okay.
Doc, do you want to pause the video real quick just to keep that so long?
Pause the video.
I'm coming, Emily.
The analogy that I would use is